Good afternoon, everybody. Today I am going to discuss about the plant cell culture and its application. So, what are application of plant cell culture? Means there are mainly two techniques. With the help of that, we are able to uh, produce a transgenic plant. One is recombinant DNA technology, in which we transfer the gene through so many gene transfer technique like agrobacterium mediated gene transfer technique, particle bombardment, that is a gene gun method through which we transfer the gene, and then liposome mediated gene transfer techniques, and then micro injection method of gene transfer techniques. So with the help of recombinant DNA technology and gene transfer technique, we create a transgenic plant after creation of transgenic cells through plant tissue culture. Simply, plant tissue culture, it is a multiplication of the plant. Means from the transgenic cells, from each transgenic cell, we can develop the each plant, and we multiply large number of plants through plant tissue culture. So plant tissue culture is simple, it is a art, but recombinant DNA technology, it is a science. Means we are going to multiply the transgenic plant. So what are application we can improve the transgenic plant means character regarding anti-insect resistant, pest resistant, weed resistance, then improve the healthy oil with altered fatty acid profile. How can you change? We change the gene that is responsible for production of fatty acid, that is not useful for us. We are going to alter the fatty acid composition, means chain of fatty acid through changing the gene level. Then modification of a starch. You know, the starch, that is the important constituent in the many syrups, many medicine, that we can improve the quality of that starch in any transgenic plant. Means we are going to produce the transgenic plant through gene transfer technique, and then we multiply that plant with plant tissue culture techniques. So we improve the starch composition in the plant as using the plant as a factory. Means it is a chip factory where we can produce huge quantity of a starch instead of using the bacteria or animal cell culture. We are selecting the plant as a model for production of that essential fatty acid or a starch. Then third point, that is the favorable change of the grain storage product and their chemical composition. So grain generally there is a deficiency, cereals there is a deficiency of lysine. So we can improve the lysine content, that is lysine is amino acid, by inserting the gene of lysine from wild species of the plant into a commercial variety. Then we can also improve the protein composition like methionine and cysteine from wild variety of the plant into a commercial variety through gene transfer technique, through recombinant DNA technology, and we multiply that plant through plant tissue culture. Next, we can also improve the quality of flour. Sticking, glutenin. Due to glutenin, there is a stickiness in the flour. So that quality can be improved with the help of transferring the gene responsible for glutenin. So we can improve the quality of the bread making, quality of the wheat flour, and then making of barley and brewing of beers. We can produce a beer from the barley, and we can produce a huge quantity, means improve the quality of that barley grains. So these are the application of plant cell culture. Means only they know single technology in case of plant cell culture. There is amalgamation of recombinant DNA technology, then antisense RNA technology, then uh, there are so many technique like creation of genetic male sterility, I have already discussed these things. Then, with the help of gene transfer technique, we transfer the gene. I have already discussed the agrobacterium mediated gene transfer, and then uh, particle bombardment or gene gun method or ballistic approach method for quick transfer of the gene from wild variety to cultivated variety. So these are the technique through which we are going to improve the quality. Means what we produce, we create a transgenic plant and we multiply that transgenic cells through plant tissue culture and produce large number of the plant cell culture plant, that is transgenic plant. We also remove the undesirable toxic compound in the cereals plant that can affect our health. It can cause allergy, it can cause disease if we are consuming directly that undesirable toxic compound in the plant like boa. Boa is a compound, BOAA, that is a chemical compound that is responsible for paralysis in human being, generally present in the legume species, Lathyrus species. That boa free seeds production that do not contain such a toxic compound that affect our nervous system. So this is the application part of the recombinant DNA technology, then plant tissue culture technology, then development of blue rose. How can we develop the blue rose? Generally, blue color pigment is absent in the rose. So we take the gene responsible for blue color pigment from any wild species plant that contain the blue color gene pigment and transfer in the rose through agrobacterium mediated gene transfer technique or through particle bombardment or through liposome mediated gene transfer technique for production of blue color rose flower. Means we can also produce the fiber that is in blue in color after transferring the gene the responsible for the blue color pigments. So this is the application part of the plant cell culture or plant tissue culture technology. Then development of tearless onion. I have already asked you a question, what, how it is possible through gene silencing. Tearless onion means a sulfur compound that cause irritation in your eye. Okay, so generally scientists have developed a tearless onion through silencing the gene responsible for sulfur compound production that coming in your eyes and cause tear. Then caffeine free coffee. Caffeine act as a blood stimulator. After taking coffee we feel relaxed because there is dilation of blood. Means we feel relaxed when blood movement fast in your artery. So there is a concept of production of caffeine present a chemical compound caffeine generally present in coffee and thein, THAI, thein in tea that is used as blood stimulator. Means flow of blood will increase when you are taking that compound through coffee or through tea. So try to remember which compound is present in coffee. Caffeine, it is a chemical compound. And then in case of tea, THIAIN, thein. So these if you are taking, then there is a quick flow of blood. So it can not only affect our nervous system, affect also our health if you are taking huge quantity. And the low nicotine tobacco. So tobacco is a model plant for production of so many chemicals, useful chemicals, and used as edible vaccine. So if you reduce the nicotine that is carcinogenic, that cause cancer. But if you silent the gene for production of nicotine compound or remove the gene that is responsible for nicotine compound, then we can easily use that tobacco for medicinal purpose. So just see the blue rose. Only it is feasible due to blue color cotton. Only due to it is feasible due to transfer of the gene inside the cotton and in the rose plant. Now, biosafety is the most important issue. Biosafety is the most important issue in plant genetic engineering. Okay, so scientists have target to feed the increasing population of the world at global level. So generally scientists have developed lot of variety of the agricultural crop that is transgenic against insecticide, insect against the pest, insect the against the bacteria, fungus, virus, and improve the quality of that seed regarding vitamin content, regarding the protein content, regarding the starch content quality. So the motto for the creation of transgenic crop is just to feed the global population, feed the animals, and fiber security means best quality of fiber that from cotton fiber, jute fiber, and other plant that is going to produce the fiber. Besides, they use in health. They also use in healthcare means production of medicinal compound that is used for treatment of different type of disease. So the constant associated with public acceptance of transgenic crop continue to be important challenging means there is some negative aspect may become due to excess use of that transgenic crop in different variety of the crops means first is the you that may cause allergy if it is causing allergy histamine secretion start there may be chance of death. So if the gm crop genetic modified crop that is the transgenic crop if produce any chemical compound that cause allergy then you never eat. So just we have to silent the gene responsible for the chemical that cause allergy. So just to test whether it can cause allergy or not. If it is causing allergy, then it is of no use. But if it is not causing allergy, then we can use easily because there is a higher production of the cereals, fruit, vegetables, fibers, fuel. So these are the application of the plant cell culture and plant tissue culture. Then effect of crop on biodiversity and environment. How it is affecting? Only single type of variety that is the transgenic crop if it is growing on whole area of the world. What happens? We are going to neglect the wild variety of the particular crop variety and that can affect the food chain and food web. So that can reduce the genetic biodiversity and due to monoculture. Monoculture means same type of variety is growing in whole world, either of the cotton crop or the rice crop or the wheat crop. In that situation, the biodiversity will reduce and it will affect the environment regarding food chain and food web. Okay, so these are the these are the negative aspect we have to think regarding the production of genetic modified crop that is the transgenic crop means monoculture crop means only one type of crop variety that is transgenic and that is growing at larger scale. Why? Because it is producing more quantity of cereals, more quantity of vegetables, more quantity of fruit, then more quantity of medicinal compound. Then farmer preferably select that crop only. And what will be the negative aspect? That is there may be chance of reduction of the biodiversity because in earlier stage in ancient time when there was no production of transgenic crop before 100 years ago we are using the variety that is coming from wild variety directly and then make a commercial variety and then there was no change in the environment then no change in the reduction of the biodiversity. Then next point is effect of gm crop on non-target and beneficial insect and microbes. What do you mean by non-target? What do you mean by non-target? Means the crop that transgenic crop there is a gene that is against the insect, against the microbes, but somehow some useful insect is coming for pollination. Means they are facilitating the production of that seed. That is the justification means. But if that insect, that microbes, that is killed by the product of the resistant gene, for other microbes, other insect to the beneficial insect. Means suppose the they gene that is resistance, that is resistant gene is transferred in the transgenic crop against other insect, other microbes, but beneficial insect is coming to the flower of that GM crop or transgenic crop. In that situation, the insect is going to killed by that genetic modified crop, or microbes is killed by that product of gene present in genetic modified crop. This is the negative aspect. Means we are going to kill also the beneficial pollinating insect and beneficial microbe that is used for nitrogen fixation, that is used for increase the availability of sulfur content, phosphorus content for the plants in the soil. Means due to presence of that microbes in the soil, the plant are able to absorb the more quantity of nitrogen, more quantity of sulfur, more quantity of phosphorus from the plant, from the soil. So this is the negative aspect, means the effect of GM crop on non-target and beneficial insect. Non-target means that insect is not targeted for killing purpose, for reducing the population of that microbes or that insect, but target is some other insect that is damaging our crop. The target is other microbes that cause disease in the crop, but that microbe, that is nitrogen fixing bacteria, sulfur fixing, phosphorus fixing bacteria, that is going to utilize. So that is the beneficial insect. Next is transgene escape through pollen. Means suppose it is a transgenic crop and somehow pollen grain is going to release either with the help of insect or with the help of wind and transfer to the weed plant. What happens? There will be development of superweed. Means the gene present in the transgenic crop, if is going to escape with the help of pollen, through the pollen with the help of insect and air, then if somehow it is going to pass on the flower of the flower of the weed, it can develop the superweed. What weed means? There is no effect of insecticide, weedicide that on that plants. Okay, so we cannot kill, even we can utilize the any insecticide. That type of development may be feasible due to escaping of gene from the through the pollen of the transgenic crop. So this is the concept of development of superweed may be feasible. Next is GM crop may change the fundamental nature of the plant as a gene from the animal. You know, the gene from the mouse and from the fish are going to introduce in the plant. So for vegetarian, from the ethical point of view, it is not purely veg materials. So that is the crop that change the fundamental vegetable nature of the plants, and it is not ethical for the society, for the vegetarian. Means if you are eating the product of the gene of the mice and fish, it means you are not 100% vegetarian. So this is the concept. Means GM crop means gene can be transferred from any microbes, uh, from any animals into the plants. So it is not ethical for the society who is vegetarian. Next, antibiotic resistant marker gene are used to produce transgenic crop, maybe horizontally transfer into microbes and exacerbate problem for antibiotic resistant in human and animal pathogen. You know, antibiotic resistant gene is going to utilize in the transgenic crop. Somehow if the gene is going to transfer to microbes and uh, others insect, that may be transferred into humans and pathogen, so that may cause the resistance in that microbes, the microbes that cause disease in the humans. So transgene may be moved from plant to gut of microflora of human and animals. So suppose there is antibiotic resistant gene already present in the plants in case of antibiotic transgenic plant, and somehow this gene is going to transfer into microbes, and the same microbes is going to affect in human. What happens? The gene, it will create a resistant. The gene will create a resistant in that microbes, and there is no effect of antibiotic on the human for killing of that microbes. In case of animal also for killing of that microbes. So this is the negative aspect. The crop, genetic crop, that is the transgenic crop, may lead to change in the evolutionary pattern. Okay, so these are the things that we should consider. First is allergy, second is the development of superweed. First is it may cause allergy, second is development of superweed, third it can affect the beneficial microbes and insect. Then it can also affect the biodiversity, means variation of the plant species, because excess utilization of same type of transgenic crop by the farmer, they are forgetting the original wild variety and affect the food chain and food web. And then we are going to change the nature of the vegetable, that is transfer of the gene from the animals and from the fish or from the mice. So it is not ethical for the vegetarian, for the society. So this is the concept we should consider. Next, development of antibiotic resistant gene in the microbes due transfer of gene from transgenic crop into microbes. So there is no effect of antibiotics on that for killing of that microbes if they are affecting the human body. So these are the concept. So this is the crop may lead to change in the evolutionary pattern, but you try to know people are you adopting the transgenic crop, that is mainly for the production of vegetable, for production of fibers, for production of cereals. Mainly the model crops are cotton, tomato, corn, and soybean. The cotton, that is useful production of fiber, that is the bt cotton. Then tomato and essence iron flavor saver. Then corn, you can produce transgenic crop against the different disease. Then soybean, roundup ready. You know already these things are utilized by the farmer after production of transgenic crop. So try to see the graphs how they are the farmers are utilizing such type of transgenic crop for maximum optimum production of that cereals, that fiber, that vegetable, that uh, fruits. So just see the uh, a record of 14 million farmers from 25 countries cultivated 134 million hectares. How many area? 134 million hectares, and that is the equivalent to 330 million acres, with the crop that are genetically engineered for herbicide. Herbicide, you know, Roundup Ready, insecticide, BT cotton, and delay fruit ripening. That is the flavor saver variety, just to get the market value of that crops and improve the oil quality. That is the unsaturated fatty acid. So these are the global area of the biotech crop planted from. This is the data of 1996 to 209. Okay, so these are the you have to learn these things. The means farmer are utilizing, and try to know what is happening in regarding the production. Okay, so you should adopt these things. You should adopt these things. Means transgenic crop, it is the utilized transgenic crop, it is going to utilize by the crop farmers for getting maximum production, for getting maximum production of that vegetables, that fibers, and that any vegetable fibers and other things. So try to know what is going on in the field of agricultural biotechnology. Okay, so you should be ready. You should be ready with this chapter for your coming class test. Okay. So, how many of you have present there? कितने बच्चे हैं आज? कितने बच्चे हैं आज टोटली बताओ? यस,
5 6
7 है.
6
कोई बात नहीं, 7 है तो देखो.
सर 7 तो
यस.
आप ही हो 6 हैं.
तो आपको नेक्स्ट डे मैं क्लास टेस्ट लूंगा. ठीक है, ठीक है. तो आपको देखना है कि ये जो प्लांट टिश्यू कल्चर है, ये मैक्सिमम चीज आप जान रहे हो. ये क्या है? व्हाट इज दिस? ये क्या है? कोई बताएगा? ए में क्या लिखा हुआ है? कैलस. बी में क्या डायग्राम है? डायग्राम दिख रहा है ना?
सिंथेटिक सीड.
डायग्राम अभी दिख रहा है?
यस सर.
हाँ, ये पहचान रहे हो ना क्या है बी में?
सिंथेटिक सीड.
लाउडली बोलिए फिर से.
सर सिंथेटिक सीड.
देन सी में क्या है? नहीं दिख रहा है क्या? डी में क्या है? डी में सोमैटिक एम्ब्रियो.
सोमैटिक एम्ब्रियो.
हाँ, अब ये डिफरेंट स्टेज है सोमैटिक एम्ब्रियो का. ग्लोबुलर स्टेज, ठीक है ना? देन हार्ट शेप स्टेज, देन टॉर्पेडो स्टेज. ये हम पहले भी डिस्कस कर चुके हैं. ठीक है. तो आप बताइए, रेड एल्गी एग्रोज का क्या सोर्स है? व्हाट इज द सोर्स ऑफ एग्रोज? व्हाट इज द सोर्स ऑफ एग्रोज? रेड एल्गी. यू नो, ट्रांसजीन एनालिसिस में जीन किसी भी प्लांट में चला गया तो इसको हम कैसे कंफर्म करेंगे? थ्रू सदर्न ब्लॉटिंग, वेस्टर्न ब्लॉटिंग. किसी भी प्लांट में जीन को आपने डाला और कंफर्म करना गया या नहीं. हाउ कैन यू टेस्ट? थ्रू सदर्न ब्लॉटिंग. ओके. नेक्स्ट
सर.
हाँ बोलो, बोलो तुम.
सदर्न प्लॉटिंग
दिख रहा है ना आपको?
सर जो आपका सिलेबस चल रहा है
क्या अभी दिखाई नहीं पड़ रहा है स्लाइड?
सर मेरा थोड़ा दूसरा सवाल है.
आवाज नहीं आ रही है
सर. सिलेबस ज्यादा
सिलेबस आपका इस चैप्टर होगा प्लांट सेल कल्चर.
एक
प्लांट सेल कल्चर.
सर मैं क्या कह रहा हूं. चलो सर आप पहले
ओके.
कंप्लीट कर लो.
नेक्स्ट है एग्रोबैक्टीरिया मेडिएटेड जीन ट्रांसफर. कौन सा डिजीज करता है? क्रॉन गॉल डिजीज. ओके. नेक्स्ट है endangered medicinally important plant can be saved through a molecular technique, that is embryo rescue. Embryo rescue क्या है? Do you know embryo abortion? Embryo abortion, it is a technique. It is problems in the rare plant species. Means embryo is going to abort after fertilization. There is no seed formation, and then what happens? Seed is not available. In that situation, in such type of plant variety, scientist is going to remove the early embryo just after fertilization. They are going to select, remove that early embryo. Okay, and then put in the plant tissue culture just to multiply that. So this is the advantage, that we are going to multiply the plant that have problem of what? That is embryo abortion. Okay. So this is the advantage. You have to use the embryo rescue. ये जो embryo rescue है, ये किस लिए यूज होता है? जस्ट टू प्रोटेक्ट द प्लांट. ये प्लांट को प्रोटेक्ट करता है. किसको प्रोटेक्ट करता है? ये प्लांट को जो कि इंडेंजर्ड है. ऐसे प्लांट को प्रोटेक्ट करता है जो कि इंडेंजर्ड स्पीसीज. ओके. सो ओनली ट्राई टू लर्न द थिंग्स the embryo rescue. What is embryo rescue? What is happen when the plant is going to abort due to chromosomal imbalance in the endosperm? What happens? There is no formation of seed, that is known as embryo immersion. But when the scientist will take the early stage embryo and put in the plant tissue culture media, then plant can multiply at large number, because there is no formation of seed through the normal methods. So this is the advantage of technique, that we can produce the plant, that there is a problem of embryo version. Okay, so try to know what is embryo rescue. Next is yes, next is father of golden iridise. कौन है? Ingo Potricus and Peter Bear. How many genes are present in golden iridise? There are 3 genes. कितना जीन है? 3 gene. PSY gene, CRT1 gene, CRT2 gene. CRT2 gene, CRT1 gene and CRT2 gene. So PSY gene is coming from daffodils, and the CRT1 gene and CR2 gene coming from Erwinia keratophora, from the bacteria, just to complete the process of formation of beta carotene. So these are, there are transfer of 3 gene. Okay, name the 3 gene, that is ये मैं बता चुका हूँ कि कितना जीन है. PSY gene, CRT1 gene and CRT2 gene. Name the 2 gene that involve genetic male sterility. आपको बताया है ना, कौन सा है? बार्नेट जीन एंड बार स्टार जीन. कितना है? बार्नेट जीन एंड बार स्टार जीन. ओके, so that is the Barnett gene responsible for degradation of the pollen grain or making the sterile. The pollen grain become sterile due to presence of Barnett gene, and Bar star gene restore the sterility of restore the fertility of pollen grain. Okay, so these are the examples you try to know. Next, that is the vincristine and vinblastine. Why I am discussing the vincristine and vinblastine? It is a secondary metabolite. ये क्या है? Secondary metabolite है. So that secondary metabolite is generally synthesized in the plant, wild plant species, just to protect the plant from itself. So Catharanthus rosea, you try to know the Catharanthus rosea. Everyone garden have Catharanthus rosea. So that contain the gene that can vincristine and vinblastine gene responsible for production of chemical compound that is known as vincristine and vinblastine, that is anti-cancerous compound. Means if one person is suffering from cancer, then it is used for the treatment for the cancer. So that is obtained from the flowering plant, that is Catharanthus rosea. You try to recognize the plant, that is Catharanthus rosea. And so many times this question has come in your CBSE exam and your medical entrance test. Then next is lipstick, that is eco-friendly lipstick. If you are using synthetic lipstick, then it can affect your health, affect your skin. So lipstick generally we obtain the naturally producing from the plant, that is Lithospermum erythroigen, that is given, that is also secondary metabolite, and that plant materials is utilized by human being. Okay, next is 2 molecular technique that used for detection of virus. कौन सा है? DNA probe technique and ELISA technique. So ELISA confirm the presence of virus in the plants, and DNA probe technique also used for the presence of virus. Means ELISA detect at the protein level after replication, transcription and translation, but DNA probe technique detect the viral gene at the DNA level. So try to know these are the names. Then why we take apical meristem for plant tissue culture to make a virus free plants? ये बहुत ही important question है. क्यों हम यूज करते हैं? Why we take apical meristem? क्यों करते हैं? Means vascular system is absent near the meristem. What do you mean a vascular system? Xylem and phloem, through which there is a flow of food or flow of minerals and water from root to leaf and from leaf to all the part, that is The pathway is flowing. So the pathway through which water, minerals is passing and food is distributing, through which only virus can transfer. But this thing is not coming up to meristem, terminal end of meristem. And you know there is a high metabolic activity in the meristem. Means and do not allow the virus. Means there is high rate of multiplication that cannot compete with that. The virus cannot compete with that high metabolic rate, and high endogenous oxygen synthesis. It will prevent the growth of virus. Means if there is a huge quantity of natural oxygen in the portion of the plant, there is no chance of infection. So it inhibits the virus multiplication. That's why we select the apical meristem for plant tissue culture as a explant. क्यों यूज करते हैं एक्सप्लांट? आपको समझ में आ गया होगा कि जाइलम फ्लोएम वहां तक नहीं पहुंचता है जो कि रास्ता है किसका? वायरस का। ओके, दूसरा कि वहां पे सबसे ज्यादा अमाउंट में जो है ऑक्सीजन सिंथेसिस होता है। इसी तरह से आपको याद रखना है, और हाई मेटाबॉलिक रेट होता है। तो ट्राई टू लर्न दीज थिंग्स। ओके, नेक्स्ट फैक्ट्स में फादर ऑफ प्लांट एम्ब्रियोलॉजी इन इंडिया पंचानन माहेश्वरी। अब एक टोटिपोटेंसी, व्हाट इज टोटिपोटेंसी? एबिलिटी ऑफ सेल टू रिजेनरेट होल प्लांट गिवन बाय हेवरलैंड इन फर्स्ट टाइम इन कैरेट प्लांट, सबसे पहले दिया था। नेक्स्ट है व्हाट आर डिफरेंट मेथड फॉर ट्रांसजेनिक प्लांट्स conformation, means gene transfer inside the plant. How can you confirm? With the help of molecular technique like PCR, southern blotting, northern blotting, and western blotting. Next example of herbicide tolerant variety, I have already told you so many times, Roundup ready. Next, a chemical that is used,
Excuse me, sir.
yes, solidifying agent. कौन सा chemical है जो solidifying agent के लिए यूज होता है? एग्रोज इंस्टीड ऑफ जिलेटिन। क्यों यूज करते हैं? बिकॉज इट विल रिमेन सॉलिड इवन अप टू 44 डिग्री सेल्सियस। सो आई विल शेयर दिस पीपीटी एंड ट्राई टू रिमेम्बर। बोलो बेटा।
सर जो सेकंड वाला पॉइंट है टोटिपोटेंसी वाला, हेबरलैंड ने तो पैलिसेड जेल में किया था ना?
कौन सा वाला?
सेकंड पॉइंट
किसका? एक्सप्लांट वाला?
हेबरलैंड ने जो प्लांट टिश्यू कल्चर किया था वो तो पैलिसेड जेल में किया था ना?
हाँ हाँ।
So टोटिपोटेंसी कैरेट में।
एक्चुअली क्वेश्चन इज नॉट क्लियर, प्लीज स्पीक लाउडली। तो एक बार फिर से बोलो। अब ये एक बार मैं ये क्वेश्चन रिवाइज करा देता, उसके बाद आप पूछते रहना। तो केमिकल दैट इज यूज फॉर मेकिंग आर्टिफिशियल सीड कौन सा है? सोडियम एल्जिनेट, कैल्शियम एल्जिनेट। वी रैप द जेल ऑफ कैल्शियम एल्जिनेट एंड सोडियम एल्जिनेट। जो आप देख सकते हो आर्टिफिशियल सीड के में ऊपर से लेयर देते हैं, किसका लेयर होता है? दैट इज कैल्शियम एल्जिनेट, सोडियम एल्जिनेट। इट एक्ट एज ए डेसिकेटर, मींस इट विल एब्जॉर्ब द मॉइस्चर। एंड कौन सा वेराइटी है टोमेटो का जो एंटीसेंस आरएनए टेक्नोलॉजी से बना है? फ्लेवर सेवर वेराइटी ऑफ टोमेटो। So these are the different. And then क्रायो प्रोटेक्टिव एजेंट कौन सा है? क्रायो प्रिजर्वेशन में बताया है ना, क्रायो प्रोटेक्टिव एजेंट कौन सा है? DMSO, वेरी मोस्ट इंपोर्टेंट। ग्लाइसिरॉल, DMSO, मैनिटॉल, सर्विटॉल, that is used as a cryoprotective agent. Why it is used? Because it will prevent the formation of ice crystal. If you put any plant cell at -196 degrees Celsius, then there is a formation of ice crystal because water is present inside the plant cell. If there is a formation of ice, then shape of volume of that ice is more than the same amount of water that is already present in the plant cell. So it will rupture the plant cell and plant cell become dead. Okay, that's why we use the dimethyl sulfoxide or glycerol or mannitol or sorbitol. So that is the advantage of using cryoprotective agent, जो कि हम दिखा भी चुके हैं। So these are the things. अब ये तो पहले भी बता चुके हैं। ऑक्सीजन साइटोकाइनिन का रेशियो 1:1 होना है तो क्या बनेगा? 1:2 होगा। तो क्या बनेगा, स्टेम डेवलपमेंट होगा, शूट डेवलपमेंट होगा। फिर जो है ऑक्सीजन साइटोकाइनिन का रेशियो 2:1 होगा तो रूट डेवलपमेंट होगा। कैलस क्या है, आपको मैं क्वेश्चन पूछा है ना, कैलस इज undifferentiated growth of apical meristem, and why we use the apical meristem as explant, I have already discussed these things. So source of barnage or RNA hydrolysis enzyme is, क्या है बार्नेज का सोर्स, बैसिलस एमाइलोलैक्टिफेसेंस, दैट इज द बैक्टीरिया, so that is the barnage. Next, try to know a strong promoter. It is the promoter only due to which the barnage gene is able to functional, that is TA29 promoter. Okay, so are you listening my son? सुनाई पड़ रहा है? Next is stress related osmolyte that has contributed, that is secreted by the plant for the adapting in adverse environmental condition for better survival of plant. उसमें कुछ जीन डाला गया है ट्रांसजेनिक प्लांट में, कौन सा, द जीन रिस्पॉन्सिबल फॉर ट्राहेलोज एंड फ्रुक्टोन प्रोडक्शन, दैट इज द शुगर, देन अल्कोहल प्रोडक्शन मैनिटॉल, एंड अमीनो एसिड प्रोडक्ट दैट इज प्रोलीन। अगर प्रोलीन है प्लांट में या जीन डाल के उसमें सिंथेसाइज हो रहा है, या बिटेन है, या एंटी फ्रीजिंग प्रोटीन है, इवन देन प्लांट कैन सर्वाइव इन द स्कार्सिटी ऑफ वाटर, इन द scarcity of water, that is in drought condition as well as in alkaline soil condition. So these are the advantage of osmolyte production in the plant, transgenic plant, means we transfer the gene responsible for trehalose and fructose production, then mannitol production, then proline production, then betaine production, then antifreeze production. So these are the stress related osmolytes. Are you listening? सुनाई पड़ रहा है? सुनाई पड़ रहा है? ये बताओ, सुनाई पड़ रहा है ना? यस, अनुष्का, अनुष्का सुनाई पड़ रहा है ना?
यस सर।
1-2 मिनट बाकी है बस उसके बाद। तो ये कौन सा है, ये डायग्राम आपको दिख रहा है, ये नॉर्मल राइस है, ये गोल्डन आयरन राइस है, जो कि जिसमें 3 ठो जीन ट्रांसफर किया गया है। वन इज PSY जीन, सेकंड इज CRT1 जीन, वन जीन, सेकंड इज थर्ड वन इज CRT2 जीन। ये है दैट इज द polyhydroxybutyrate that is synthesized by the plant Arabidopsis and Populus plant, that is biodegradable. That is the gene transfer in the plant, that is in Arabidopsis and Populus from the bacteria. Okay, so these are the globules that is responsible for production of means biodegradable plastic, that is polyhydroxybutyrate. So these are the diagram. नेक्स्ट ये क्या है आपको पता है, मैं बहुत बार पूछ चुका हूँ, माइक्रो प्रोपगेशन का मेथड है। ये लैब में ग्लास हाउस कंडीशन में, ग्रीन हाउस कंडीशन में, प्रोटेक्टेड कंडीशन में ग्रो हो रहा है। सो दीज आर द थिंग्स दैट इज इंडिकेट, एंड यू सी दिस इज द क्रॉन गॉल डिजीज, दैट इज ड्यू टू disease due to the bacteria agrobacterium tumefaciens. You will find that is the gall like structure you will find,